Hi, my name is Helen Tai. I'm a research scientist with Agriculture and Agri-Food Canada. I'd like to thank the organizers for giving me this opportunity to talk about our work on genome sequencing of adapted diploid potato clones. Current commercial potato varieties have complex autotetraploid genetics that hamper improvement through breeding. Potato also has a high heterozygosity that masks deleterious alleles. In this schematic diagram, you can see the genetic combinations of diploids and autotetraploids, and you can see that it's much more complicated genetically using tetraploids for breeding. So this leads to the question, how about breeding potato as a diploid crop? Diploid potato is already used for cultivation and breeding. For thousands of years, farmers in South America and North America have cultivated several diploid species that produce tubers in addition to Solanum tuberosa. There are over 100 cultivated and wild species that produce tubers. Most are diploids. Some species can induce tetraploids to become dihaploids, and these dihaploids can then be used for diploid breeding. Diploids have been developed at Agriculture and Agri-Food Canada, or AAFC, where I am located, including release of a variety AAC candy cane. These diploids were developed through outbreeding. What is being discussed more recently is diploid inbreeding. Diploid inbreeding will require self-compatibility mutants. So these self-compatibility mutants will confer selfing to diploid lines to generate inbred lines. However, self-incompatibility occurs in most diploids, and generation of inbred lines will require the use of these self-incompatibility mutants. There are many advantages to diploid inbreeding. The inbred lines need to be selfed for 6+ generations before use in F1 hybrid breeding. F1 hybrid breeding has the potential for heterosis or hybrid vigor. Also, deleterious alleles are purged with inbreeding and beneficial alleles are fixed. There's more effective use of marker-assisted selection, reduced complexity in genetic crosses, as we discussed earlier, and true potato seed can be used for rapid propagation in commercial production, as opposed to the use of tissue culture in generating nuclear stock for current tetraploid production. We had a number of legacy diploids from the retired breeder, Dr. de Jong. These legacy diploids have a background of introgression with wild species, and 9 of them will be discussed here today. And here's the picture of the flowers of the 9 diploid clones that we'll be talking about today. The diploid lines also showed a lot of variation in traits, including tuber dormancy, as you can see here in one of the studies that we've done. Dr. de Jong also looked at a number of traits, including vine maturity, chip quality, some disease resistance, eye depth, skin color, and a few other traits. But mostly what Dr. de Jong did was he selected for good agronomic traits and tuber quality traits and fairly good tuber yield. The tuber yield is as high as 70% of the tetraploid tuber yield. Many of these clones have been deposited in the US Potato Gene Bank and our SP6 in Sturgeon Bay. Genome sequencing of these diploids was done using the 10x Chromium platform with de novo assembly. 10x sequencing is droplet-based sequencing. So within a droplet, you can get assembly of a single haplotype. And so with the different droplets, you can get assembly of a single haplotype. However, to get the entire phased genome will require additional sequencing. Nevertheless, with the Chromium platform, we're able to determine things such as heterozygosity, which is very important for our study. So we use the genome sequence information to predict deleterious alleles using the algorithm SIFT. And SIFT uses the genome sequence to predict protein sequences, and from these protein sequences, there is an algorithm that predicts the functionality of that protein. Different genome sequences, reference genome sequences, are needed to be used with the SIFT algorithm. And in our study, we found that there were differences in deleterious allele calling depending on the reference genome. And here you can see a figure showing the total number of deleterious alleles. The DM is the use of the DM reference genome and the RH reference genome was done. And you can see that there are differences between them. The green is the DM, and the blue here is the RH, and yellow is DM, and dark green is RH. So there's differences depending on the reference genome. Each clone also shows differences with other clones, and there's differences in the number of heterozygous and homozygous deleterious alleles between the clones as well. So the 9 diploid clones show differences in patterns of heterozygous deleterious alleles across the genome, in addition to the total number of deleterious alleles. And you can see here, these are the 9 different clones that were studied. The total number of genes is indicated by the blue bars, and the percent heterozygous deleterious alleles is the green lines, and this is the bins of genes across the genome for each of the 9 clones. So you can see that, and this is just showing chromosome 12, and you can see that for some clones there's very few heterozygous deleterious alleles near the centromere, at the center of the chromosome, but for others there's a widespread distribution across the chromosome. We were very interested in heterozygous deleterious alleles because in inbreeding, these deleterious alleles can become homozygous and have the potential for lethality. We're going to focus today on discussing H412-1. This clone was derived from W4, which carries the SLI gene that confers self-compatibility. We tested the H412-1 clone using the Casper marker that was originally described by Corentin Clow in 2020 in Theoretical and Applied Genetics. And from our Casper genotyping, we determined that H412-1 is a heterozygote for SLI. You can see that the deleterious alleles analysis shows widespread heterozygous deleterious alleles across the genome for this clone. Zooming in on the location in the genome where SLI is, we find that there is an adjacent gene that carries a heterozygous deleterious allele. This heterozygous deleterious allele can be problematic, and recombinants that show a separation of the SLI allele from this deleterious allele will be selected amongst the progeny. However, alternative strategies may be needed since the heterozygous deleterious allele is very close to the SLI locus. And so a CRISPR-Cas9 strategy, for example, may be required in order for us to get rid of this deleterious allele. So we were also interested in the ST-CDF1 locus since it confers long-day tuberization, and homozygosity of this allele would be advantageous. We looked at chromosome 5 where the ST-CDF1 gene is located, and you can see here again there is widespread distribution of heterozygous deleterious alleles across the chromosome. Zooming in on the location of ST-CDF1, we find that there are 2 regions flanking this gene that carry heterozygous deleterious alleles. Again, recombination between ST-CDF1 and these loci that have the heterozygous deleterious allele will be needed. Or, employing a strategy like CRISPR-Cas9 is also possible. So in summary, Potato can be diploid or tetraploid, and diploid F1 hybrid breeding has some advantages over current tetraploid breeding for genetic improvement. Generation of inbred lines will require self-compatible diploid potato. Analysis of genome-wide deleterious alleles was done to identify regions that should be selected against in the development of diploid inbred lines. I'd like to thank my collaborator at McGill University, Martina Stromvik, and Sai Achakagary and Maria Kyriakidou in her lab, who contributed to the de novo genome assembly and genome analysis. In my lab, I'd like to thank Charlotte Davidson and Emily McCoy, who were involved in working on these diploids. I'd especially like to thank Henry DeYoung, who generated these very valuable diploid lines, and Kyle Gardner and David DeCoyer, scientists at AAFC that worked with me on this project. This was funded by the AAFC Genome Research and Development Initiative, as well as Compute Canada and Genome Quebec. I'd be happy to take questions. Thank you.